Something with this.
So we can start, I think. Fayçal, we can start with you. Let me introduce Dr. Zeroual Fayçal. He's a lecturer in veterinary medicine at the University Shadil Baneth and Tark. Sorry for the pronunciation. And expert in veterinary parasitology and vector-borne diseases, reproductive pathology, artificial insemination, and genetic improvement in cattle. And then he provides us with a state of art of zoonosis in North African countries. The floor is yours. 20 minutes.
Thank you. I try to respect the time. Thank you very much, Mr. Chairman. Okay, I will present you the state of art of zoonoses in North African countries. We are to focus on the countries who are borders with in North Africa with border with the Mediterranean Sea. Want to work? No, wait. Yeah. Okay. For the introduction, the countries are located in south of the shore of the Mediterranean basin and made of 5 countries: Morocco, Algeria, Tunisia, Libya, and Egypt. This region is very dynamic subcontinent in demographic term, with a population that exceed 2 million people and living with the most worrying health of and education problems globally. North Africa countries are vulnerable to several transboundary animal diseases due to their geographic location and their borders with the Sahel zone. North Africa also is linked to Europe by great economic mobility, commercial exchanges, legal and illegal human movements, as well as the free movement of migrators also bears. All of these factors effectively contribute to ongoing transmission of numerous infection and vector-borne disease, and consequently favor the appearance and reappearance of emerging and re-emerging diseases. We start with zoonoses caused by bacterial origin. First, the brucellosis, who is also named indolent fever or Mediterranean fever or Malta fever, is one of the major infection worldwide. According to the World Health Organization, more than 500,000 new cases of human brucellosis are reported each year worldwide. However, it's estimated that the number of undiagnosed cases in 4 times higher. For the Egypt, this pathology is endemic, and it was described for the first time in 1938. The prevalence of human and animals brucellosis is for 1.25% for the human. In Algeria, we have 80 cases for 100,000 inhabitants. More than 100,000 are confirmed cases of animal brucellosis. In Morocco, the average of 2.2 5 cases per year of brucellosis cases in Morocco were reported. In Tunisia, we have 100 cases, and in Libya, 50 cases are reported. For the tuberculosis, also, it's second global in 2020. This second was the second global leading causes of death from an infectious disease behind the COVID-19. For the tuberculosis in Egypt, it estimates an incidence rate for 10,000 per year. In Algeria, we report 42 cases per 100,000. In Morocco, the first this disease is the first cause of death, and the number of cases in Morocco is around 13 cases. For Tunisia, the incidence of tuberculosis is also declining and went from 20 inhabitants in 2008 to 22 cases in 2021. The disease in Libya is estimated for 4,000 cases and occurring in 2021, and the rate tuberculosis-related death in Libya is 3 persons per 100,000 per year. For the rickettsiosis, who are the vector-borne disease by vectors, Haematophagous arthropoda, we start with rickettsiosis. These rickettsiosis are caused by Rickettsia genesis with strict intracellular bacteria, and they constitute the group of infectious disease transmitted by vectors like lice, fleas, ticks, etc. We show here the rickettsia, the most rickettsiosis in form in North Africa. We have the Mediterranean spotted fever caused by Rickettsia conuri, tick-borne lymphadenitis by Rickettsia slovakia and Rickettsia rauti. We have also the Rocky Mountain spotted fever caused by Rickettsia rickettsii, and African tick fever by Rickettsia africae. The most serious being exanthelmic typhus or epidemic typhus. It's caused by Rickettsia prowazaki, and the main vector is the lice. For the borreliosis or Lyme borreliosis, it's zoonotic disease caused by Spirochete Borrelia bergdorferi and transmitted primarily to humans through the bite of infected Exodus ticks and other hard ticks also, like soft ticks and lice. In Egypt, it has been detected and isolated from animals, breeders, and also human. Also, we note in Egypt there is another species of Borrelia like Miyamotai and Afzeli. In the rest of countries of North Africa, the genesis of Borrelia are also found in hard ticks, especially in Exodus ricinus, and in soft ticks. Human cases in Algeria, Morocco, and Tunisia of Lyme bears were diagnosed in patients in whom the disease manifests itself by facial paralysis and neurologic and joint signals. Another pathologist, anaplasmosis, in this region of North Africa. A few dozen publications have reported the presence of anaplasma in human, dogs, horses, cattle, sheep, goats, camel, and only in hard ticks. In hard ticks. For the Q fever, who is caused by bacteria Coxiella burnetii, is transmitted by ticks genus Exodus. Naturally infected animals, the man here is an accidental host. The chronic form of Q fever is virtually identical to endocarditis because it caused the inflammation of the inner lining of the heart and causes abortion also. In the countries of North Africa, the seroprevalence of antibodies reactive with Coxiella burnetii in these countries were similar to those reported from the southern of Europe. It was in several animals also, species, also in human cases. For the zoonoses of viral origin, we have first rabies disease, which caused with lysaviruses, and it was always fatal as soon as clinical symptoms appear. She is endemic, and in most cases, the rabic virus is transmitted to humans throughout the saliva of unaffected animals, domestic or wild, particularly dogs and cats. Also, the wild carnivores can play the role of transmission. They are also the reservoir. Each year, about 60,000 fatal human infections are reported in the world. In the Egypt, we have 30 to 40 annual cases. In Algeria, around 20 cases. Morocco, 50 cases human death. And in Tunisia, also from 50 to 20 deaths. In Libya, the number is underestimated, and the first case was reported in bovine rabies in 2018. For the Rift Valley fever, who is a viral zoonotic zoonosis that primarily also affects animals, but also affects humans. It's transmitted by mosquitoes and the flies. It was reported among livestock in Valley Rift, Kenya in 1900, and illness range from the mild flu like illness to severe hemorrhagic fever that can be fatal in humans. It causes significant economic losses in livestock like mortality and abortions. In Egypt, we have report 27 deaths in 2018, and the virus antibodies were detected in 14% of veterinarians and their assistants, butchers, and abattoir or slaughter workers in pigs abattoir. In the rest of countries, we have never reported any cases, but only a priority disease for this disease is in the Maghreb, composed from Morocco, Algeria, Tunisia, and Libya, due to the threat of introduction of the virus through transboundary livestock movements and infected also mosquitoes vectors. For the West Nile fever, the virus is arbovirus here and mainly transmitted also by mosquitoes that causes the neurological damage. Migratory birds here play the main reservoir to be the reservoir of transmission. Since we have the birds are mostly subclinical, and they are responsible for widespread and reintroduction from zoonotic to new regions. For the Egypt, in the Egypt, West Nile fever is circulating the virus in circulating in the most or virus various region of Egypt and causing febrile illness. It causing up to 24% of human serum sample were positive. In Algeria, we have 6 outbreaks of West Nile virus infection involving equines in 2023. And one fatal clinical was reported in Algeria in the region on the border of Mediterranean in 2012. Morocco, the West Nile was only detected in equine. In Tunisia, we have unfortunately 723 confirmed human cases of West Nile fever, with 31 unfortunately deaths. And in Libya, West Nile also is detected in horse and was reported in by serology. For the dengue fever, who is also viral disease and spread also this disease by mosquitoes to people. It's more common to tropical and subtropical climates. In Egypt, we have reported 2 cases of this disease. Also, we report, the 2 cases of dengue of virus in Italy, 2 cases who have been reported from South Egypt. The same year, in 2011, was reported the reappearance of Aedes aegypti. The cases of dengue fever were reported also in Moscow, returning tourists, and 6 additional Belgian, Australian, and German from Egypt. In this year, the health department of the Red Sea Governorate reported the dengue fever outbreak, and so between 1,200 and 2,500 people were declared infected in Egypt. In Algeria, we report only the vector, Aedes albopictus mosquito, and the disease without any human cases. In the Morocco, also the vector was detected with the presence of an imported cases from African country like Cote d'Ivoire. And in Tunisia also the mosquito was detected, and the vector competence was also confirmed by experimental infections. In addition, vector competitions with chikungunya and dengue and Zika for this vector with Aedes albopictus. For Libya, neither disease nor vector have been reported until now. The Zika virus, since the discovery in the Uganda forests, the Zika virus has spread to several African countries and Asian, and it causes congenital malformation, prompting specific vigilance for pregnant women in the event of Zika epidemic. In Egypt, the virus has spread between the first decennial of the last time. Even though no Zika cases have been reported, and the vector mosquitoes is present in this country. For the rest of the North country, Africa, this region border to the endemic countries like Egypt and sub-Saharan countries, have not reported any cases, but it's necessary to remember the presence of Aedes albopictus is present in these countries, and that the verification of vector competence has been confirmed by experimental infection with this virus. Crimean Congo hemorrhagic fever also, it's born disease. Animals like domestic, like wild, and also birds and cattle, sheep, play a role in the life cycle of the ticks, as well as in the transmission and the amplification of the virus, without showing clinical things. The disease was declared twice in Mauritania, bordering country, North African countries. In Egypt, a surgical study provided the first evidence that antibodies to fever hemorrhagic Crimean Congo were present in several wild and domestic animals, including camels, ruminants, sheep. It's several species of ticks, especially genesis of Yaloma. 3 cases of this disease have been identified in 1981 in Egypt. In Tunisia, the virus have been reported in cattle, sheep, also goats and ticks, and slaughters, and in slaughterhouse workers also. The study or several studies carried out in Tunisia also reported the detection of this virus in ticks collected from dromedaries. Dromedaries ticks generally is Yaloma dromedaries genesis. In Algeria and Libya and Morocco, these countries bordering endemic countries also have not reported the case. But it's therefore urgent to develop a solid strategy to combat this risk and spread the virus in these countries using one health approach. Now for the zoonotic parasitic origin, we start. We have a serious problem with leishmaniasis. Cutaneous leishmaniasis, for example, in Algeria is in class in the rank 4th rank in the world. The reservoir there is cutaneous leishmaniasis. We have several rats, wild rodents, and also the visceral leishmaniasis, who is fatal in more than 95% cases in left and untreated. We note that in Algeria we have not the third form cutaneomycosis, only in South America and other region. The Hydatidiose is very dangerous, also zoonotic disease. It's reported with highly percentage in human, in several animals, also in pigs only in Egypt, but in wild birds worse, like Algeria. For the malaria, this is very big or dangerous disease with 290 million people infected. We know that 95% of people infected are origin in Africa. We note also the dangers of the second vector with Anopheles stephensis, who is coming to spread also in from the Horn of Africa to another countries. This year we report also the presence of stephensis in so in Ghana, in Nigeria also, and maybe in another countries. We have to verify. For the toxoplasma also is a very important. Okay, just I see the name of the disease, toxoplasmosis, and the last with the fasciolosis is very neglected zoonosis, and it's very spread in the region of Basin Mediterranean. Man can contract the disease by eating some plants like cresan and pisali. For the zoonotic fungal in North Africa, the research are very small, and we want to improve the research in this. D'accord. Thank you for I just if you want to for the conclusion, understanding the dynamics of disease between animals and human. We have to elevating the burden of zoonotic disease in the region. Throughout prioritize the research, prevent measures and work in one health approach. And thank you for your patience and for assistance.
That was an immersive experience in all North Africa teams. Any questions for Fayçal? Yes, Alessandro.
Sorry, maybe I didn't get the Rift Valley cases that you have in humans. I saw that 14% are vets, but then butchers, and I was surprised about
In Tunisia?
Yes, people working in big abattoir.
In Egypt, yeah.
Yes.
In Egypt.
Yeah, no, because I was reading some papers in which they did experimental infection with the Rift Valley fever virus in pigs. And it seems that they get infected, but they don't really spread. So this is a very interesting observation, because I didn't know that pigs, no, not myself. So yeah, so is there any publication on this, or maybe some data? Because
in Egypt, yes.
Okay.
Yeah.
So it's
in Algeria, no. In Algeria, we have تفضل.
No, just sorry, I didn't get your.
If I don't start in Egypt, they are slaughtering pigs.
Yeah, it was surprising also to me, but also this big spreading Rift Valley fever, because I don't know. Thanks, but this is something that I didn't know.
This is the yes. There is also research published the research in this one.
Any other question? Thank you, Alessandro. Any other question? Just a very quick one, Fayçal. There is a lot of diseases surrounding Algeria, but not many there. So, what is the active surveillance in your country? What are the major virus or pathogens that they are looking at? Is there any active surveillance?
For the surveillance, yes, but it's not sufficient. It's not sufficient. For this subject, we have to prepare a lot of teams who can, let me say, start really the surveillance and also the epidemiologic studies for several, because there is a lot of work to do in no, not only in Algeria, but in the exactly, yeah.
Thank you so much, Fayçal. Thank you. We can go to the that's so YouTube. We go with the Jairo. Correct me, perfect. So, Jairo is assistant professor at the University of Bari at the Moro. Recently, he has studied the Leishmania species of reptiles.
No, it's
And
for the
mammals
point.
in areas endemic for Leishmania infantum and the Mediterranean basin. His presentation is full of special effects.
Hopefully.
So, so if you are feeling to sleep, this will pump you up. And look at that. Sorry, I have 20 minutes.
Okay.
As lovely as you are.
No worries. Thank you so much. Good afternoon to everybody. And thank you so much also for the opportunity to talking about one of my favorite group of animals. Unfortunately, very disregarded as a group of animals that can have a very important role in the transmission of zoonosis. So I will take these 20 minutes to illustrate to you some examples of how reptiles have an important role within the transmission of zoonosis and how we have to have an active surveillance and include them in the concept of One Health. So as a veterinarian, the way we try to keep the health of these animals is through the, start working again. Let's see. It's through the herpetological medicine. Herpetological medicine is that area of veterinary medicine which includes the health of these animals. It's a wide area, so I have been focused in the last few years in parasitology, which is my main area of action or research, and the approach within the One Health approach and to assess the zoonosis these animals can have. With this, we have the other concept, which is herpetological parasitology. In this case, we have to consider that these animals, as diverse as they are, they also include a lot of different types of parasites in this specific case. This is me many moons ago. So when you consider reptiles, as you have to consider not only the diversity they have as animals, but also individual diversity of parasites. So when you have an animal that you have to examine, you have a wide diversity of parasites. Most of them are already have a convergence evolution with this type of animals, so they are non-pathogenic, and they also are already symbionts of these animals. Some of them can be very pathogenic to reptiles, and some of them can be zoonotic. So to study the zoonotic parasites of reptiles, we have to consider how they interact with humans, because we're used to think or the general belief is that reptiles are very far away in the jungle, but actually we interact with them in a daily basis in the very different contexts and epidemiological and societal concepts, such as, for example, the industry or the group of people that have reptiles as pets is growing exponentially, specifically in the northern hemisphere, but also many regions are used to having herpetophagy, which means feeding on different type of reptiles, not only as the specific cuisine, but also as a way of perpetuating some traditional behavior. But my area of research has been the synanthropic relationship we have. What is this and why this is so important if we consider the Anthropocene and the environmental modifications? The Anthropocene is the era we are in right now. And in this era, the anthropic pressure, which means, for example, urbanization, deforestation, and fragmentation has created that practically animals that were first forested, now they're moving towards urban areas. You can see here the projection of Italy in the next couple of decades. It's practically the whole peninsula will be an urban area. If we have urban area, animals have to go somewhere. So animals will go to the cities, and then we have an increased synanthropy. If we have an increase in entropy, that means that animals are getting used, they are plastic, they are adapted to those type of environments, and they can thrive. And if we have those animals interacting with our domestic animals and even humans, we will have the increase of encounters. This means we will have not only accidents or, for example, predation of these small animals in different contexts, but we also will have the increasing interaction between humans, domestic animals, and reptiles, which means an increase in the transmission of pathogens, and in this case, or in my area of expertise, parasites. I will do a short paragraph on this part, or a short part of this to explain something that is very important. We're talking about neglected tropical disease. And one of the most important neglected tropical disease are snake bites. Snake bites represent not only a high burden for in tropical countries with thousands of deaths per year, but also a lot of sequelae or consequences to people that are having this problem, which has not have the particular interest or investment in this area. But if we look at the other side of the picture, venomics and also the identification of compounds of these venoms have helped not only in the discovery of new drugs, but also, for example, in the treatment or control or in the effective effect these drugs can have for parasites such as malaria, leishmania, or trypanosoma. So when we consider, coming back to this talk, we consider zoonotic diseases. When we think of reptiles, we think of Salmonella. Generally, we think that reptiles, and this is true, reptiles are all asymptomatic, and most of them can have some type of zoonotic Salmonella. But if we go back to thinking on parasites, what we have is that we have a myriad of parasites that can be transmitted through different routes. With this paper, we try to identify, or we identified 3 typical or major ways of transmission. So it's through herpetophagy, or feeding on, eating raw or uncooked meat of reptiles. The other is environmental contamination, and the one that we are working more, which is vector-borne pathogens, not only parasitic, but also bacteria and virus. So if we consider reptiles within that paradigm of what are zoonotic pathogens, they can have 2 functions or 2 roles, which is paratenic or intermediate hosts. For example, when we talk about helminths, and they can be also a definitive host, such as, for example, pentastomids, which are a crustacean-like zoonotic parasite, and also mites and ticks. So this is very important, considering what I talked before regarding domestic animals being able to predate. And indeed, dogs and cats that have a outdoors lifestyle, they not only contribute to the decrease or the decline of populations, but they also have a zoonotic impact, not only bringing those animals to our homes, but also getting infected. For example, with Mesocystoides, which is a cestode that can only also be infected for dogs and cats, but it's also a typical or a paradigmatic example of zoonotic parasites. Other zoonotic parasites that have been studied in the recent years, including reptiles as paratenic hosts or intermediate hosts, are Spirometra, which is another type of cestode related to water environments. And in this case, what you can see here is that already it's been many studies confirming that this parasite is also occurring in Europe. In this sense, it's associated to snakes. It can be also seen infected as humans as intermediate hosts. And the dissemination or distribution of these pathogens is through predation, specifically from cats, because they are the definitive hosts. Whereas if we talk, for example, of snakes or reptiles as definitive hosts, we can see the example of pentastomids, which they are very pathogenic to their specific host. They can even kill when the adults, and they are very pathogenic not only to the intermediate host, but also for their specific hosts, such as, for example, this is the largest viper in South America, Lachesis muta, that died because of these parasites. And when we enter in contact, not only different type of mammals and even humans, to the eggs or larvae, we can have pentastomiasis. Pentastomiasis is a high burden disease in tropical countries. And we recently described these adults from the lung of geckos in the southernmost part of Italy transmit, and this is the larvated egg of this type of parasites. And we describe this species coming from Linosa Island in the southernmost part near to Tunisia, which probably could be a new parasite that can have a zoonotic concern, considering that it can be transmitted or arrive to the continent through predation and through cats. So this is the example I wanted to give regarding the transmission of contamination or predation, but we also have our focus on vector-borne pathogens, and this is our main area of research in the more recent years. So reptiles, they can be reservoirs or hosts of 2 specific type of vectors, which are acarina or mites and ticks, but also dipterans such as mosquitoes and sandflies. So of course, they can have a very direct effect on their hosts, and this will obviously be related to the amount of parasites infesting, but also parasites are very intelligent and they have different strategies. And even if you look at the mouth of a snake, you can find more and more different type of parasites adapting to different environments, such as the mouth of snakes in Central Europe. Two examples that I want to give regarding vector-borne pathogens is mainly bacteria. I can talk about recent studies we've been performing regarding Lyme disease and Mediterranean spotted fever. The first was in Basilicata in the southern part of Italy, in which we described many types of ectoparasites from reptiles, but we have a special focus on Ixodes ricinus, which infested a lot of different synanthropic lizards, and also Neotrombico autumnalis, which larvae also infest, mammals. Then we wanted to give a more holistic approach, a more One Health approach. So then we went and studied in the same area for Rickettsia and other Rickettsiales. And what we saw is that not only reptiles, but also ticks in their environment and humans were exposed to a myriad of Rickettsiales, such as Rickettsia conori, Rickettsia monacensis, and Rickettsia helvetica. So actually reptiles, they can have a very important role in the circulation and perpetuation of not only enzootic cycles, but also zoonotic cycles within people that are exposed to this type of parasites, for example, ticks. Another example is still in the part of the vector-borne mites. We can talk about how hepatological medicine and parasitology can contribute to the control of some parasites. So, for example, this is the snake mite, which is Ophiuchus natricis. It's a worldwide distributed mite. It's hematophagous, and it feeds mainly on snakes, but it can be zoonotic, so it can also feed on humans that are in contact with these snakes. So in Sicily, recently we found a highly infested place where we found not only snakes that were kept for breeding, but also a private collection. And what we did was we tried to eliminate or eradicate these parasites through a normal drug used for dogs, which is anisocosaline called afoxolaner. So we got all the snakes that we assessed, and then we treated them orally. Some of them given the pill through the mouth, and some of them through the prey to be able to eradicate completely. And we took blood to see if the pharmacokinetics of the drug was the same as in mammals. And actually, it's the same. So we were able to eradicate completely and not only protect the snakes from this environment, but also humans that were in contact with these animals. The last example I want to give you regarding vector-borne pathogens and our very, let's say, important research we're performing is Leishmania. So for those of you that you don't know, Leishmania has, of course, the pathogenic Leishmania, but also has a sister clade called Sauroleishmania because it's associated with lizards mainly. And Ceriatomya minuta is the main vector of this type of Leishmania, 21 species more or less. Sauroleishmania, indeed, some of them can infect humans. This is a case of Leishmania adleri in Kenya from Latasia longicaudata, this type of lizard, which actually can produce cutaneous leishmaniasis. If we come back to the Mediterranean basin, what we can see is that we have the the sympatric occurrence of different species, such as Leishmania infantum, of course, the canine leishmaniasis agent, but we also have Leishmania tarentulae, which is, if you have never seen it, it infects the Moorish gecko, Tarentula mauritanica, that's the name, and it has a peripilarian type of development, so it's able to infest the stomatal valve of Ceriatomya minuta. What's interesting about this parasite, it's actually in the recent years, we've been seeing that it can infect mammals. So we have seen some infection in humans, serologically and molecularly, but also in dogs. So dogs being the main reservoir for Leishmania infantum, we wanted to see if it could also infect them. And what we observed is that not only it can be positive molecularly, but serologically, which brings us a great opportunity, but also a big hindrance. Considering this, and we went to see if Leishmania tarentulae and also infantum, they were also having an important role within reptiles. So we did a study within the 4 main regions of the southern part of Italy. And what we saw is that, yes, both species infect reptiles, at least if we look at them molecularly. And then we did, in the recent years, different studies, for example, in Marrakesh, where we saw Leishmania tarentulae in other type of reptiles, such as chameleons and snakes. And more recently, we did some studies in central Italy and southern Italy, and we saw Leishmania tarentulae in many types of snakes, for example, the four-line snake and the western whip snake. And we saw Leishmania infantum within the bone marrow of some geckos, and we were able to isolate Leishmania tarentulae from geckos. So you can see here that we further confirm that possibly these animals can have a role within the circulation of Leishmania infantum. But importantly about the isolates is that we were able to infect derived monocytic cells from dogs, and we were able to see that in fact they can persist in these cells, which means that it's not only a transient infection, it means that they can create an infection in these animals. And then most importantly, recently, we evaluate the cytokines, and what we're able to see is that they produce a Th1 response, which means that they could be used not only as a candidate for vaccine, but as an immune therapy, which is our goal in the next couple of years. So this is the recent distribution of Leishmania tarentulae, which infects different type of host mammals and reptiles. And our epidemiological scenario, at least in Italy, is this. We have the Mureta seco, we have the lizards, sandflies, and canine leishmaniasis, as well as reptilian leishmaniasis or reptilian leishmania. There's still many things to do. We know a lot about Leishmania infantum, about Leishmania tarentulae. There's still many gaps in its transmission, in its biological cycle. So we are working on this. But to finish, I would like to give you two examples of our recent concept in which we're trying to include not only the interactions humans have with reptiles, but how the cultural interactions we have with reptiles can be studied under a One Health approach. So it means ethnoherpetological or ethnoherpetoparasitology, which is related to different contexts, for example, festivals, the snake charmers, the medicinal purposes of reptiles, and also predation of domestic animals. One example is in Morocco. We did a study in which we went to assess the animals that were kept in the souks of Marrakesh. And what we wanted to see if they have different pathogenic or zoonotic pathogens. I will give just two examples because it's a long list of pathogens, but we saw Anaplasma phagocytophyllum in snakes related to, and also Midichlorian midichlorian, which is an endosymbiont, meaning that they were infested priorly with ticks. And we also found some species of Rickettsia belonging to the spotted fever group. On the other hand, recently we are collaborating with this type of studies in which we include parasitology, One Health, but also conservation. Considering that we are in the Anthropocene, we are causing these animals to become more and more synanthropic. One example of this is the Festa dei Serpari, which is a ritual, sacro-profane ritual performed in central Italy, in which they use four-line snakes to adorn the statue of San Domenico. It's a more than 500 years ritual, which has a very important, not only cultural, but traditional connotation. So we studied not only the parasites, but also the relationships humans have and the positive effect maybe these type of events can have not only on humans, but also on the snake population. And we found some ectoparasites that may be new. We're still working on that. We did the first coprological study on wild snakes, seeing many different types of parasites, some of them spurious parasites. And we also, of course, found some reptile vector-borne pathogens such as Rickettsia. So we're still working on that. And the whole goal of this type of studies is not only to assess these animals and include them in the One Health approach and how they contribute to the transmission of zoonotic pathogens, but also how can we elicit or create policies for their conservation as they are part of our environment. With this, I want to thank you for your attention.
Thank you so much, Jairo, for two things. You finished on time. That was fantastic. The second one, the presentation was amazing. I mean,
Thank you so much.
Sticker. I will give you a sticker. It was a fantastic presentation. So
I was running on coffee, so that's why I'm so.
Okay, any question for Jairo? So I will ask you something. It's a very stupid question. I know that people will say
There's
it's a
no
stupid question, but this is very stupid. But You touch a bit at the end, but I still don't understand what is the real impact of these pathogens in Southern Europe. I mean, it's possible to quantify how much disease they are involved.
So it depends on the effect. Yeah, for example, regarding the bacterial Lyme disease and rickettsia, they are, at least in some cases, they can have a very important role because they are the ones that are keeping the larvae and nymphs of Ixodes ricinus, which is the main vector for those type of bacteria. And in that specific case, when we went to work in a park in Basilicata, we saw that the people that were working there, they were already exposed. And the main, if you go to do a carological study in that area, you will find that the main reservoirs or hosts for Ixodes ricinus are lizards. And they are also positive for those type of pathogens. So they are keeping that context. The other part is that not everything has to be bad, in the sense that, for example, regarding Leishmania, what we're seeing is that maybe the infection with Leishmania tarentule in mammals could produce a natural immune response that will, let's say, a natural vaccine that will allow infected animals with Leishmania tarentule to be able to respond or have a polarization to a Th1, which means a cellular response, and then they will be protected against an infection with Leishmania infantum. And we have seen, I think maybe Domenico is going to talk about that. We have seen that, for example, we have dogs. Generally, the paradigm for dogs is that you have high antibody titers. The dog will, in a couple of months, begin to have a clinical appearance of the disease. But we saw and we monitored dogs for 1 year, 2 years, and they were having extremely high titers with any clinical signs. And then we saw that they were also infected with Leishmania tarentule.
Okay, fascinating. Thank you so much.
Thank you.
Any other question? No? You need a second round of applause. Thank you so much. For the speaker for today and for the last plenary. So the last speaker for today is Baneth, Professor Baneth. Is a Ribac Pearson Chair in Veterinary Medicine. Among the many national and international roles that he did in the Professor regard, he covered in his career. He was a chairman of the World Small Animal Veterinary Association, Scientific Advisory Committee. Professor Baneth is involved in the study of zoonotic veterinary diseases in the Mediterranean basin, Uzbekistan, Ethiopia, and Southern Europe. He's author of more than 300 scientific publications and book chapters, and served as advisor for the European. He served as advisor for EFSA, which is the European.
With this. You press this. Not on here. Don't we have our remote control? Okay. We can use one from that.
Okay.
Okay, good.
While we're getting the remote control, I'd like to thank the organization and the organizers of this conference for the invitation. And I've been given a very difficult task, I'd say impossible task, of describing the role of pets in the transmission of vector-borne diseases. So we're just going to touch the tip of the iceberg in this talk, 20 minutes, maybe less. And I'll try to just talk about some examples of diseases, but make sure that it works.
Just check here, maybe it's, I turn it on. Yeah, I turn it on, sorry. Okay.
Okay. No.
Okay, maybe.
Okay. So just and that's one. Okay. So let's just talk first about pet animals in Europe. And apparently there's an estimated 145 million dogs and cats living in Europe. And this was in 2022. And the numbers are increasing. So just by 5 million compared to 2021. 66 million dogs and 79 million cats. Here in this map, we can actually see where there are more dogs and where there are more cats. The red is more cats. And this is compared to a total population of humans in Europe of 447 million humans. So roughly about one pet per every 3 people. And if we could estimate that perhaps a family had 3 people in it, so then one pet per family, more or less. Just to show you the importance of the number of pets that are present. And if we look at other animals, in this case, livestock, we can see here the numbers per year, and we can see that the pig population in light blue is more or less stable. However, the bovine population is decreasing, and so are the sheep and goats over the years. That means that we're going toward a smaller population of domestic production animals, while the number of pets is continuously increasing. And this is also to show you the importance of pets and the fact that they are the closest animals to us, so they live with us, not like farm animals. They are actually in our bedroom, sometimes in our bed, and we have a very close contact with dogs and cats. So in general, if we talk about zoonotic diseases and not just vector-borne diseases, of course, dogs and cats harbor and transmit some very important zoonoses like rabies, like toxoplasmosis for the cats as the main reservoir, and echinococcosis, and these diseases have been mentioned and will be probably mentioned more and more during these 2 days. And when we talk about vector-borne diseases, there are several very important vector-borne diseases that are associated with pets, like leishmaniosis, also of cats, not only of dogs. And we know that recently there are more and more studies describing this infection in cats and disease caused by leishmania in cats. So this is becoming much more important than we thought 20 years ago. And of course, the dogs are still considered the main reservoir host in most countries. We'll also consider diarrhea-filariasis or diarrhea-filariosis with diarrhea repens and imitis, and I know that will also be mentioned in the next talks. Rickettsiosis with different Rickettsia species like Rickettsia felis, Rickettsia connori, and in the Americas, Rickettsia rickettii. And there are also many other different Rickettsia species that are associated with infection and disease in people. Borreliosis has also been mentioned. So we're talking about Lyme group Borrelia, like Borrelia burgdorferi sensu-lato, but also about relapsing fever Borrelia, which are different and are a different cause of disease transmitted by soft ticks. And I'll mention them toward the end of my talk. So, these are some of the vectors that we can see here and you've heard about. And when we speak about animals, and especially pets, as reservoirs, we need to think about whether they are really truly a main reservoir or are they accidental host or secondary reservoir. And I'll get to the definitions of what these are in the next slide. So we can definitely think about leishmaniosis and diarrhea-filariosis as diseases in which probably dogs are the main reservoirs. However, in Lyme disease with Borrelia burgdorferi and in tick-borne encephalitis and in other infections, some animals are just accidental hosts and not really reservoirs. Let's speak a little bit about Leishmania and Leishmania infantum. We've heard a little about this infection and the importance, one of the most important zoonotic diseases in Europe and globally. And we can speak here about different types of hosts. So the primary reservoir host is a host that actually maintains R naught higher than 1. That means that the ability of one animal or one human to infect another individual is higher than infecting just one, because if it's below 1, then the disease should diminish with time. So we've heard with coronavirus, a lot of us got to know the concept of this, and this is really an important point. A secondary reservoir host is a host that can transmit infection but can't maintain the transmission cycle in the absence of the primary host. So I'll show the example of dogs and cats with leishmania, and probably without having the dog as a main reservoir, cats would probably not be an important reservoir for infection to other animals with leishmania infantum. Accidental reservoir hosts are just hosts that can be infected, but usually don't transmit the parasite in the case of leishmania. With any disease, we should think about just infected hosts, so any animal that may establish infection, and those that are truly reservoirs. And what is a reservoir, or what is required from an animal to be a good reservoir for leishmania? And I'm not talking just about leishmania infantum in this case. It's also other species of leishmania like Major tropica and the American species. So the animal should be available to sandfly vectors, because we know the sandfly vectors are actually the vector transmitting the infection. They should be abundant. They should be infected at a high rate. They should be very attractive and infectious to sandflies, and they should be able to maintain a year-round infection. So an animal that has a very short lifespan and doesn't last the whole year is probably not going to transmit the infection further in the next years, and we will need an animal that is able to live until the next sandfly season in the case of the old world, because that's not the case in the new world, where we know that actually leishmania is being transmitted by sandfly year-round. So if we look at the primary host cycle here, this is the dog, and we know that actually dogs are considered a main reservoir host for leishmania infantum. In the case of the cat, if the cat is truly a secondary reservoir host, and we still need to study this further, but I'm not aware of any area where there are just cats without dogs and transmission of leishmania infantum. And there have been some anecdotal reports of other animals like hares, and perhaps on Monte Cristo Island also rats, but not a situation where cats have really been shown to be the only reservoir hosts. So most probably in the absence of the dog, cats will not be a good transmitter or a good reservoir for leishmania infantum. In many places, also wildlife are infected with leishmania infantum, and they can also participate in the life cycle. However, again, here we think that they're probably not a main reservoir. And we know that in South America, for instance, leishmania infantum was not established before Columbus, before the Spanish and the Portuguese brought their dogs there and infected sandflies and so on. So this is, of course, not proof that dogs are the only reservoir. However, it's probably the situation. When we speak about an endemic area for leishmania infantum, we can think about a population of dogs which are susceptible to infection. They will most probably meet the parasite by sandfly bite at some stage, or probably an early stage of their life. Some of them will have good immune response and may not continue to become infected or continue to be infected. However, a large part of the population will maintain persistent infection and a very long latent period, usually with no clinical signs. Most of these animals will remain subclinical carriers of the disease, and only about 10% will eventually develop clinical disease. However, this part of the population is also able to infect sandflies, so they also participate in the epidemiology of this disease. When we look at the clinical manifestations of leishmaniosis with leishmania infantum in dogs, we have a wide variety of clinical signs, and this starts with papular dermatitis, a very mild form that is present in dogs, for instance, in the Bizen hound and on the Balearic Island. And these dogs have been selected for years and years and years, have gone through natural selection and don't really manifest a clinical disease due to this infection. They have these small papules that are often seronegative, and they can be PCR negative in the blood for leishmania, and usually they recover spontaneously. Unfortunately, this is only a very small part of those dogs that really show clinical signs, and others may develop a very devastating and severe disease, which often ends with renal disease and death. So if we look at the population of dogs in an endemic area, we have this classical pyramid with a very small tip of dogs that have clinical signs, and under it, a lot of infected dogs that can be seropositive or PCR positive. This is like a tip of an iceberg situation. This is a fairly recent study that had looked at leishmaniosis due to Leishmania infantum in Europe, and has actually approached all the ministries of health in every country in Europe, and also the veterinary authorities. And this is a map that was made to show where infection is the most prevalent in red here. And here are the numbers of dogs that have been reported to be infected to the authorities. So in total, we have between 2015 and 2020, a total of 4,183 dogs reported as having leishmaniosis. This is, of course, not representing the situation at all, because even if you look at the published scientific research papers, you find a higher number of dogs that have been described to be infected. So there's a severe underreporting of leishmaniosis in Europe, and also, by the way, of cutaneous leishmaniosis in many other areas of the world, including the south of Europe. Let's continue to another disease. So deer filariasis, I already mentioned, and Dirofilaria repens is apparently emerging and is also reaching new areas in Europe. And this is the current map of where it's present. So we've been used to knowing that it's actually in the Mediterranean basin. And right now it has also reached the Baltic area, so the Baltic countries, and it's apparently continuing to progress and reach new areas. And this could be due to global warming or to changes in the climate, and perhaps also to other factors. So this is the disease that is not so pathogenic to dogs, but when we speak about Dirofilaria immitis, the cause of heartworm disease, we also find a situation where it is also becoming much more prevalent than it was before. And here, this is also an infection that is zoonotic and can cause pulmonary manifestations in humans. However, to the dog itself, it's detrimental and it causes terminal heart disease. What about borreliosis? So Lyme disease Borrelia with Borrelia burgdorferi and other species apparently can infect dogs, and but it looks like the dog is just an accidental host here, and actually the infection revolves between rodents and ticks that feed on deer, and this is the situation in North America and apparently also in some parts of Europe. And although dogs are often infected, and if you look at the seroprevalence of infection in areas of North America and some areas in Europe, you find a very high seroprevalence, but they are only dead-end hosts and probably are not contributing much to transmission to people. Relapsing fever Borrelia. This is a group of Borrelia that is transmitted by soft ticks, Ornithodoros species and others. And I found in Israel, my home country, that actually dogs and cats are infected with a species that causes a severe disease in humans. It was not known before. And they manifest signs like fever, they have thrombocytopenia and anemia, and it could be a fatal disease to them. And you can see here blood smear from a cat and a blood smear from a dog, and you can see how common the Borrelia is in the blood, and this is a situation which you don't see with Lyme Borrelia. In Lyme Borrelia, it's very, very rare to see a spirochete in the blood. So this disease is common apparently in cats and dogs, and the question is, are there reservoir for humans? Could it be that they are related to the epidemiology of the disease in humans? So we did a very large study to look at ticks and to look at the blood meals of ticks that are found in caves, because that is usually where people are infected, and apparently also to look at wildlife. And apparently there's a very high infection rate in wildlife, and most of the ticks have a blood meal from several mammals, but not from dogs and cats. So apparently the situation here is quite similar also with dogs and cats, similar to the one found with Lyme disease. Cats and dogs are infected. They may suffer disease, but they're probably not important in the epidemiology of this disease. So just to summarize, I've given some examples of diseases where dogs are main reservoirs and cats are main reservoirs. However, we should be able to carefully distinguish between those infections in which dogs and cats and other pets, like even those pets mentioned in the previous talk, are probably important reservoirs, and the situation where animals are infected. They may even have disease, but they're not really an important part in the epidemiology of the human disease. So just to go back to the beginning, dogs and cats are the animals that are the closest to us. The numbers are growing while the numbers of other livestock are actually going down in Europe, and they are infected with a large number of different pathogens. I just touched really the upper surface of what I could speak about here because we have a limit of time. And I think that the emphasis should be given on what to do to prevent the disease. And I, of course, didn't have time to speak about it, but I think probably Domenico Pranto will address this in his talk. So thank you very much.
Thank you, God. I really appreciate it that you stay on time. Sorry about that. You had a big task to do. Any question for God? Questions?
So
Yes, please. Yes.
May I ask you, thank you for this overview about all climate-borne disease of zoonotic concern and other zoonotic diseases associated with dogs and cats. May I ask you, what do you think in the future, which are among those that you described, the vector-borne diseases that are probably at an increase in terms of prevalence, also considering that we have the isoptosalines and repellents that are largely used in many countries in the Mediterranean basin.
Yes, good question. So you're asking me to be a prophet?
Yeah.
And I think that what we're seeing right now is an uprising in the diarrhea-filariasis situation, where it's entering more countries in Eastern and Northern Europe and reaching territories where it probably wasn't before. And this is despite the use of ectoparasiticals and so on. And I suppose that this trend might continue. With regard to Leishmania, the awareness of owners and veterinarians is growing higher. And I think that we will probably see some stabilization. And I would expect that if it enters new countries, then there will be outbreaks. For instance, there are some studies that have shown that it's actually entering the south of Germany as well. And over there, people are not so aware of it and probably not using the right protection. So probably could be outbreaks in areas like that, especially if global warming brings a warmer climate to those areas where there's an interface between where the infection is present now and where it could further go.
Any other question for God? Okay, thank you so much, guys. Patricia, is the coffee machine working now? Okay, so we have, let's do 15 minutes break, and then maybe we do 40 minutes networking, will be an open session, so we will not do groups, because we are not many today. And then around 15, 20 to 5, we can go back, because I can see a lot of people is tired, long travels, but let's stop for 15 minutes and then we start again.